My name is Madalena, this is Robert, and today we are here to show you our screen. Cancer affects all of us. Almost everyone in this room knows someone that has faced this terrible disease. Scientific research has led to the development of very important therapies. However, cancer is still common, deadly, complex, and one of the greatest unmet medical needs. And this is due to two reasons. First, due to the resistant invasion to current treatments, and second, but very important, due to the disease complexity and its fast progression, which makes very important and crucial the discovery of new targets so that we can now unlock the next generation of cancer therapies. And this new target is R loops. As you might remember from your biology classes, our DNA encodes in our genes our genetic information. Our genetic information is then read through a process called transcription, where a new molecule that carries now this genetic information continues to pass this genetic information, and this is called the RNA molecule. Sometimes the RNA molecule can bind to the DNA of the gene and forms a structure, an entanglement, called an R loop. And it has been observed that R loops, when are elevated in the cells, can sometimes lead to cell death. Importantly, these high R loop levels are usually seen in cancer cells. However, cancer are viable and they can still survive. But now, if we target these R loops with drugs that increase even more the levels of these structures in our cells, we can target cancer cells because these R loop levels will reach a toxicity tolerance and the cell can no longer cope with these levels and will lead to cell death. And this can happen due to two mechanisms. Either, as I showed you before, high R loop levels lead to genomic instability and cell death, or, very importantly, increased R loop levels can lead to a cancer cell visibility. And now, the immune system can understand and detect cancer cells, activate an immune response, and in this way lead to cell death. And we can now improve immunotherapy. And the role of R loops as the next target is being supported by multiple key opinion leaders. But until now, it was impossible to screen for drugs targeting these structures. And why is that? Because there was no sensor to screen for them until we came along. Back in the lab, we have developed a tool called RHINO. RHINO is composed by a binding domain that simply binds to the R loops, and here in our RHINO, we also have a fluorescent marker. And this means that now, with the RHINO, we can label R loops and we can see these structures under the microscope as these green dots. RHINO is a proprietary R loop sensor that offers high specificity and sensitivity. And it's also scalable for high-throughput screening imaging assays that can be used for drug discovery. Our screen, our team, we use a very simple workflow. We are based on RHINO cell models, then are in multi-well plates, and with our automated system, these cells are incubated with drugs. And then here, these cells are taken to the microscope, and we can detect which drugs increased R loop levels. And now, currently, we have a capacity to screen 410 compounds in 24 hours. And what we offer? First of all, we offer uniqueness. We are the only screening platform for R loops. Secondly, we offer speed and efficiency. We can now test drug libraries to identify molecules that increase R loop levels. And, very importantly, we offer specificity. The new drugs identified can have a precise effect on cancer cells. And we have proved our screen through the use of approved drugs in cancer therapy. We are placed in a high-throughput screening market, and here we will focus on the oncology-focused drug discovery segment, which brings our market to $4 billion. Our business model is twofold. First, we will offer commercial screening services, where biotech and pharmaceutical companies can provide us with their libraries, we screen for them, and then we give them a full report saying which drugs increased R loop levels. Second, and very important, we will also focus on drug licensing. We will buy libraries that are publicly available, we will screen, and then here we will perform identification and validate, and then IP protect these new drugs that we can identify. We started with the pipeline development, and currently we are performing assay validation and quality control by performing pilot screenings and define standard operation procedures. And now we plan on scaling up and develop our business, and for this, of course, we need to attract investment. And we plan to enter the market regarding the commercial screening services in 2027, and for this, we seek an investment of 1 million euros. On the other side, for the drug licensing, we start in the same way, but now here, we plan to start screening available libraries by 2026 and receive our first milestone payment by 2029. And for this part, we need an investment of 2 million. The investment needed will be used to acquire automated systems, and then we will, by 2027, when we enter the market with our cursor screening service, we will generate our first revenue by a rate per plate. And then, at the same time as we are going with our drug licensing pipeline, by 2029, we forecast the first milestone payment of 75 million, and this is data from two proxy companies that have a similar path to ours. And then, as we grow and expand our company, we will be able to generate more revenue. The team behind this project is Robert and I, and we work at GIM. And I want you to take this message home. Cancer is very complex and heterogeneous. So there is a constant need for new targets and new therapies. But now we can align a new target, which is our loop, with our tool, RHINO. And in this way, our screen will open the door to a new class of cancer drugs. And thank you very much.